right, everyone, it's week 2. We are talking about cells this week. So hopefully this is a lot of review for most of you, but we are also going to go into a bit more detail about certain things that you may not have covered in previous classes with Chapter 2. Alright, so our introduction here just kind of covers our learning objectives. So we're going to talk about structure and function of the cell membranes and how it regulates movement of materials through that cell membrane, so in and out of the cell. We're also going to talk about functions of the different organelles that are contained within a cell. Talk about structure and contents of the nucleus, which is really important. We'll talk about the cell cycle and steps of cell division in our somatic cells. So somatic cells are the ones that are not gametes, the sperm and egg cells. And then we'll talk about cellular differentiation, how we get super specialized cells for different tissues and different organs. This video, we're just going to focus on the cell membrane. The other videos will cover the other sections of the chapter. So for the cell membrane, we are going to go into detail about the molecular components of the cell membrane, major features of that membrane, and then talk about materials that can and cannot pass through that lipid bilayer, and then different types of transportation that can move other substances that cannot diffuse through the membrane. Alright, so really quickly, the cell membrane is made out of something called a phospholipid bilayer. We'll talk more about that in a second. It also has cholesterol and a lot of embedded proteins in the membrane. And so the overall function of the cellular membrane, also sometimes called the plasma membrane, is that it is a protective barrier around the cell. It keeps all of the cellular components like the cytosol and organelles inside of the cell, and it keeps other things outside of the cell. And then it is also going to help control what materials can get out of the cell and what materials can get into the cell. And that's where that cholesterol and embedded proteins really come into play to assist with that regulation process. So a phospholipid is a molecule made out of a phosphate head, as shown here in this kind of reddish color. So this is phosphate with some glycerol attached to it. So the phosphate head is called hydrophilic. Hydrophilic means that something is attracted to water. So this is friendly with water. It can attach to water, no problem. And then we have 2 fatty acid tails attached to that phosphate head. All right, and these are hydrophobic, which means that they repel water. So lipids are fats, and so if you think about mixing oil and water, it makes a lot of sense why these would repel water as well. So this phospholipid bilayer is a bunch of those phospholipid structures lined up side by side, and then back to back as well. And then we have 2 layers of that are facing in opposite directions, as you can see here. And so in one direction we have phosphate heads. This is the extracellular area, so outside of the cell. So we've got those water-loving heads, the water-friendly heads, facing outside of the cell, and then we also have heads facing the internal part of the cell, or the intracellular area. And then those hydrophobic tails, those fatty acid tails, are going to be attracted to each other kind of in the middle of that bilayer. So bi means 2, so we have 2 layers made out of these phospholipids. All right, so let's talk a little bit about membrane proteins now. So we have 2 main types that are very commonly found in cell membranes. These are integral proteins. These are embedded directly in the membrane. So inside of these phosphate or phospholipids, so kind of crammed in between some of these. These include channel proteins and cellular recognition proteins. And then the other type is peripheral proteins, and these are going to be found entirely on either the inner surface or the outer surface of that membrane, and we'll see some examples of these in a minute. So this is a diagram showing what a cell membrane actually looks like. And again, this is just a graphical representation. So we have our phospholipid bilayer here with those phospholipids in 2 layers, and then you can see a bunch of different structures embedded in and on the surface of this membrane. And so we can see these channel proteins in here that pass all the way through both layers of the phospholipid membrane or plasma membrane. So we've got proteins that pass all the way through, and then we've got these other proteins like this peripheral membrane protein over here and right here that, as you can see, are only attached to one of the 2 layers. All right, and there's some reasons for that. It's based largely on what are those proteins doing, what is their function, what substances are they transporting around. So we'll go into more detail there in a little bit. And then we've also got some glycoproteins. These are these proteins that have a carbohydrate tail attached to them. And then we also have glycolipids. These are lipids or fats that have a carbohydrate attached to them. And those have their own purposes as well. We're not going to go into a ton of detail about those though. Plasma membrane also has some other extensions or structures. So first up, there are microvilli. These are very, very small folds in the membrane that are going to increase the surface area. We talked a little bit about these last week when I was discussing structure and function of the small intestine. So we do have microvilli on our small intestine to help increase that surface area so that we can absorb more nutrients. Next we have cilia. These are little hair-like structures that can produce movement. We see a lot of these in our respiratory system among other areas, but in the respiratory system, those hair-like structures, the cilia, are going to help move mucus around, and the mucus traps pathogens and other debris like dust particles and stuff in it, so it's not getting deep down into our lungs ideally, and these cilia are going to kind of move in a wave-like fashion to move that mucus along so that it can get checked by our immune system to see if they need to take care of any pathogens or anything and clean that up a little bit, and basically keep that mucus out of our lungs so that we are not getting all of those substances. And we also don't really want mucus in our lungs a whole lot. A little bit's okay, too much is not good. And then we have flagella. These are tail-like structures that can also produce movement. The only cell found in humans that has a flagella are sperm cells. So we don't see flagella very much in the human body. We see it a lot more commonly in bacterial cells, which again, we're not talking a whole lot about in this class. So this is kind of what some of these structures can look like. So over here on the left, we see these microvilli. So these are folds in the surface. Again, we saw this last week with the small intestine. Here in the middle is a representation of cilia. I know this diagram makes it look very similar to the microvilli, but they are different structures with different purposes. So the microvilli are not going to produce any sort of movement, so they are just folds to increase that surface area, whereas the cilia are going to move. So like I said in the last slide, they're going to kind of move in like a wave-like fashion. So if you imagine tall grass blowing in the wind, that's kind of the movement that the cilia are doing. And then we have our sperm cell over here with the flagella. We'll talk more about that later in the semester when we do the reproductive system. All right, let's talk about transport across the cell membrane. And so this is going to involve a lot of those membrane proteins that we were talking about a couple slides ago. So the cell membrane or plasma membrane is selectively permeable, and that means that it is able to allow some substances to pass through, but prevent others from passing through. So it is selective in what it allows through. So it does this through a couple different types of transport, passive and active transport. So passive transport is going to be movement through that membrane, either through proteins or directly across the membrane itself, without needing to use energy to make that movement happen. And this occurs because these materials are going to move down their concentration gradient, which means that the substances are going to move from an area of high concentration where there's a lot of those particles, and they're going to move into an area of low concentration where there are fewer particles like that, and it's going to keep up that movement until there's roughly equal concentrations of the substances on the two different sides of that membrane. This process is known as diffusion, and we have, as you can see, a few different types of diffusion. So we have simple diffusion, facilitated diffusion, osmosis, and filtration. Simple diffusion is going to look at substances, really small molecules. This could be something like some salt ions or potassium ions. Sometimes it depends on what cell we're talking about. Other cells, those do have to use other types of transport, but other things like glucose can also happen here because we really need glucose in our cells, so we need that to be able to get into the cells very quickly and easily. And so as you can see, starting out here, we've got the extracellular fluid up here at the top. Intracellular fluid is going to be down here, so inside of the cell. So if we start out with a lot of these small molecules outside of the cell, so there's tons and tons outside of the cell, there's none inside of the cell. Slowly, over time, these molecules are going to move through that phospholipid bilayer, and they're going to keep moving again from this high concentration area to the low concentration area until there's roughly equal numbers between the extracellular fluid and the intracellular fluid. All right, so this is just a slow, gradual process of movement directly through that plasma membrane without the assistance of those proteins usually. Facilitated diffusion is going to use some of these proteins. They're going to use channel proteins. The idea is still the same, that we are moving from an area of high concentration to an area of low concentration, but in this case, these molecules are going to be things that are too big to just pass directly in between these phospholipid structures, and they need a little bit more space to get through. And so we have these channel proteins. So this is basically just like a tunnel that goes through the membrane, allowing these larger molecules to pass through from outside of the cell to inside of the cell, or vice versa. All right, but otherwise it's still going to be that diffusion process where we're moving from an area of high concentration to an area of low concentration, and that's going to keep happening until we have roughly equal concentrations. Sometimes there are also carrier proteins involved in this process that are going to help kind of guide these molecules through those tunnels and assist them in getting across. But that is facilitated diffusion. So these channel proteins are facilitating, they are helping the movement of these larger molecules. Now we are going to look at osmosis. So osmosis is diffusion and movement of water molecules specifically. So water is so important and so unique that it has its own special type of diffusion that it undergoes. It's still a similar idea. We're moving from an area of high concentration to low concentration, but water is a pretty unique molecule. It has some odd properties that are a little bit different than what most things do. And so we are still looking at that semi-permeable membrane. This dashed line is representing that plasma membrane, that semi-permeable membrane, and this is slowly going to move around. So in this diagram, the water is represented by this kind of pale red, orangey colored substance here, whereas other molecules are these little purple dots everywhere. And so as you can see, what is occurring here is we have, starting out in this image, equal amounts of water on either side of that semi-permeable membrane, but there's a lot more of this other molecule on one side than the other. And so what's going to happen here is instead of these molecules moving around, usually this is going to happen when these molecules are too big to get through that membrane, is that the water molecules are going to move. And it's going to move towards that area where there's a lot more of those other types of molecules, because the water's trying to equalize the concentration of that molecule on either side of the membrane. And I know that's a little bit different than what we just looked at, but if we look at this image over here, you can see that we've had water move from the left side of that membrane to the right side of that membrane, and this makes the concentration of those other molecules equal. All right, so if you were to calculate out how many of these molecules there were by a certain quantity of water, those concentrations are going to end up being equal. All right. Osmosis is a really important concept because of how much it affects our cells. So I will show an example of how that can affect certain types of cells in just a minute. So osmosis, again, this is occurring when there's an imbalance of what are called solutes, those other molecules. So these little purple structures are solutes. This can be ions or bigger molecules and stuff like that. So we have some different types of concentrations of these solutes in this solution that have special names. First is isotonic solutions. This is where the concentration of those solutes inside and outside of the cell are equal, so there's no net movement of water. And that net movement basically means that water molecules are still moving back and forth through that membrane, but there's not more water going in one direction than the other. So we're not measuring more water on one side than the other because the movement is equal back and forth for an isotonic solution. A hypertonic solution, so this is when concentration of solutes inside of the cell is lower than outside of the cell. So hyper, that prefix means above, so that concentration of solutes outside of the cell is higher than inside of the cell. So our net movement of water is going to be moving it out of the cell to try to equalize those concentrations and make the concentrations inside and outside of the cell to be isotonic. All right. And then we have hypotonic solutions. Hypo means low. So the concentration of solutes outside of the cell is lower than what is inside of the cell. In this case, the water is going to move into the cell. Lots of water is going to move into the cell in an attempt to get that to be an isotonic solution. So, if we're looking at this with human cells, we're going to use blood cells in this example. This is a really important thing. So, if we've got an isotonic solution, as seen here in the middle, that's great. That's what we want. So equal amounts of water and solutes moving in and out of these red blood cells that are pictured here. So they're maintaining their shape appropriately, which means there's plenty of space for hemoglobin to carry oxygen around the body. These cells are functioning exactly the way they're supposed to. If we come over here on the left and look at a hypertonic solution, so the concentration of solutes is higher outside of the cell, and again, we're going to try to move water out of the cell to try to equalize that concentration, and as we pull water out of these cells, the more water we pull out, the more they're going to shrivel up, which means less space for hemoglobin, less room for oxygen molecules, so these cells are not functioning the way they're supposed to inside of the body and are able to carry oxygen and carbon dioxide and stuff around to where it needs to go. All right, and if this keeps going, these cells will eventually die because they just collapse in on themselves and there's just no space for anything and there's not enough water around. On the flip side, over here on the right, a hypotonic solution, so concentration of solutes outside of the cell is lower than what's inside of the cell, so our water is going to move into the cell to try to equalize those concentrations. And there's only so much space inside of a cell. Those membranes are only going to stretch a little bit. So the more water we try to move into these cells, the fuller they're going to get. So imagine trying to blow up a balloon and you just keep adding more and more and more and more air into that balloon, and it's going to pop eventually, right? If you're not relieving that pressure somehow, that balloon is going to pop because there's simply too much pressure on the plastic outside of the balloon. Same thing happens to the cells. There's just too much pressure on that membrane. It is going to rupture and burst, and the cells will die as a result of this. This process is called lysis, so the bursting apart and breakdown of cells. So having either a hypertonic or hypotonic solution, as you can see, is really not great for us. All right, and this is part of the reason why, if you're ever given an IV in some sort of medical setting, or you are going to become a medical professional and you are giving IVs, these are special solutions, so you'll frequently see them called saline solution or lactated Ringer's solution. There's a couple other types out there, but those are two of the most common ones. And you want to be giving people those saline or Ringer's solutions because they are isotonic solutions. So they have a concentration of solutes that is going to match what is normally in the human body, so that we are maintaining that water balance and we're not risking somebody being put into that hypertonic or hypotonic situation, and that is going to destroy a lot of their cells, not just the red blood cells. That's just the example being used here. But this is why we don't give people pure water. All right, this is also why if somebody's very, very dehydrated, again, they could be given an IV. Sometimes we use oral rehydration solutions. And again, that is not just plain water, because that is going to produce a hypotonic solution. It's going to decrease the concentration of solutes outside of the cell because we're adding more water in, but no additional solutes with it. And so these rehydration solutions are going to have electrolytes in them, like salt and potassium and stuff like that, to make it an isotonic solution so that we're not risking either of these scenarios on the ends. All right, let's move on and talk about active transport. So active refers to the fact that it requires energy to move materials. In this case, we are moving materials against, not with, their concentration gradient. So we are actually going to move things from a low concentration to a high concentration rather than the opposite with passive transport. All right, the most common types are protein pumps, and then endocytosis and exocytosis. So sodium-potassium pumps are used in many areas, many different types of cells in the body, muscle cells, nerve cells especially. Sodium-potassium pumps are really, really important. And so this is using those membrane proteins and energy to move different molecules, in this case sodium and potassium, in or out of the cell. And again, we are moving against the concentration gradient. So if there's a lot of sodium out in the extracellular space, as you can see here, there's very little sodium in the intracellular space, but we need that sodium to be at a higher concentration outside of the cell in order to trigger certain reaction processes. We'll talk more about exactly how that works when we get into muscle contractions and nerve impulses and stuff later in the semester. This is just kind of a general introduction to these different types of transport. So we are trying to move more of the sodium outside of the cell. And on the flip side, we are trying to move potassium into the cell because we want different concentrations, because As you can see, sodium and potassium, they have these little plus signs next to them. That means that they have a positive charge to them, and that is going to create something called a membrane potential. That basically kind of gives a head start to some energy for certain things to happen. All right, and so if we move all the sodium to the extracellular space, we have a big positive charge out here. Potassium in here is going to have also a big positive charge. Depending on the cell, we need different concentrations in different places. I'm not going to go into detail about all of those situations right now because it'll make more sense when we're talking about each unique situation this is used in. But we need to kind of get some of these molecules moved in and out. And so as you can see, these membrane proteins, we are using energy, this ATP, that's what's giving us energy. So these sodium ions, they're going to move into that space in the protein. We're going to have some ATP come attached to that protein. That is going to trigger the bottom of that protein to shut and the top of it to open, allowing those sodium molecules out into the extracellular space. And then we can also add a phosphate back on from ADP, adenosine diphosphate, and do this process in reverse with the potassium ions. Let's talk about endocytosis now. So we have 3 types of endocytosis. So endo means inside of, so this is a process where we are bringing something into the cell using an engulfing process. So it's kind of like the cell is eating these substances with the membrane. All right, so we have phagocytosis, pinocytosis, and then receptor-mediated endocytosis. All right, but in all of these 3 different types, we are bringing something into the cell by creating these little packets with the membrane. All right, so phagocytosis, this is used for very large particles. Lots of pathogens, viruses, and bacteria utilize this method. Phagocytosis is also performed by various immune cells. Pull these pathogens in so that they can destroy them. And so these are, again, very large particles. And so as you can see, the cell is going to basically start creating this little pocket with its membrane, and it's going to keep moving that up until we have completely enclosed that large particle, and it's going to seal it off. So it's this nice little packet. In phagocytosis, it's called a vacuole, and that little packet contains whatever that large particle was, and then the cell will, basically these other 2 ends of the membrane are going to seal shut and reconnect to each other. And pinocytosis, similar process, smaller substances though, and otherwise same idea. We are going to basically create a pocket. In this case, instead of reaching out for those substances, we are going to, as you can see, kind of dip in to create that pocket, and then still pinch that off so that we end up with, in this case, a vesicle is what it's called in pinocytosis, and then that membrane will reseal itself together. Last but not least is receptor-mediated endocytosis. So we are using a receptor to help with this process. All right. And so what is occurring here is the receptor is necessary to trigger the membrane to start creating this pocket. All right. So the substance that's trying to get into the cell is going to attach to those receptors, kind of like a lock and key situation. That is going to signal to the cell membrane and these little molecules here on the inside of the membrane that, hey, this thing is trying to get into the cell. As long as it looks legit and not dangerous, the cell is going to go ahead and proceed with that process of engulfing that particle and create, in this case, a coated vesicle. So we've got these extra structures on the outside. We'll talk more in detail about different situations where this process is used later on in the semester. Exocytosis is the reverse process of endocytosis. So we're taking these little packages of something, and those are going to fuse with the cell membrane, and then that membrane will open up to the extracellular space and release the contents of that little package, and then the membrane reseals itself up. All right, so exocytosis is just the opposite of that endocytosis. Endo means into, exo is out of. All right, so not too difficult there, I don't think. No subtypes of exocytosis that we're going to talk about. This one's pretty straightforward, thankfully. And that wraps up the first section of the textbook. In the next lecture, we'll talk about cytoplasm and cellular organelles.